Reshma, I don't know if I can sit awkwardly silent for another full minute. Why don't we start
We
with
can chat. We can
why don't
chat.
I start with the
So,
most boring introduction? I can introduce myself. Yeah, well,
Well,
while people are joining, Robert Blaget, director at NBC BioLabs, we're a collection of incubators here in the Bay Area, home to nearly 100 companies. Remarkably, BioLabs companies have more than 54 programs in the clinic. Combined, those companies have raised more than $7 billion. And we're really a place to help entrepreneurs get their company off the ground and get started. And so if you're in the audience and you're thinking about starting a biotech company, maybe this fireside chat will help inspire you to do so. And if so, we've got a lab bench for you. With that, maybe Reshma, would you introduce yourself?
of course. And everyone, we'd like to get a sense of who's here as well. So if you want to put your LinkedIn or website in the chat, I'll start. We can kind of get a sense of who's here. But I'm Reshma. I am grateful to know the folks at NBC BioLabs, as I was a alum in the past and worked in the lab. And I'm currently a visiting partner at Y Combinator. So we do early stage company investing. And a lot of people don't know this because I think YC is mostly known for its software investments, but we do invest in a lot of bio companies. And if you're interested to learn more, I will paste a few links here about some of the companies who've gone through the program and some of our writing and materials on the topic. And we're similar to NBC BioLabs, really hoping to encourage people to take the first step in starting their company and think about themselves as potential founders. So, let's see, it's 11:04, so why don't we start with the program? It is my pleasure today to introduce Nancy Pryer, who is a translational scientist, experienced in building and leading teams, advancing targeted therapy from discovery through exploratory clinical development and in oncology and rare diseases. And, we're talking about AmbAgon Therapeutics today. Nancy is currently the Chief Scientific Officer at AmbAgon. And it's an early stage company discovering and developing small molecule stabilizers of protein-protein interactions as novel oncology medicines. So, and they're both Nancy and Michelle, who will be introduced soon, will tell us more about the science and the IP of the company as well. And prior to AmbAgon, Nancy has held executive positions at Day One Biopharmaceuticals and Neurix Therapeutics, and led translational sciences at BioMarin, as well as scientific teams at Onyx Pharmaceuticals, Sugen, and Novartis Institutes for Biomedical Research. So a lot of experience in the field with a lot of top companies here as well. And she's delighted to have collaborated with many stellar colleagues to discover and develop 8 marketed drugs benefiting patients in need, including, I practice this at home, hopefully I won't botch it, Albosolib, is that right? For patients with HR+ and HER2 metastatic breast cancer. So it's my real pleasure to introduce Nancy, and of course, we're here today to talk about the founding story of AmbAgon. And Robert, you're up
Yeah,
next.
so Reshma and I will be trading off this interview. I have the honor of introducing Michelle Arkin. I actually will not try to say any of the drug names because I'm always terrible at it. But Dr. Arkin is professor and chair of the pharmaceutical chemistry at the University of California, San Francisco, and she is a co-founder of AmbAgon. Her lab focuses on developing drugs and probes, particularly with an interest in protein-protein interactions and protein degradation, as you'll hear more about today. Michelle holds several leadership roles at UCSF. She helps run the Small Molecule Discovery Center, the Academic Drug Discovery Consortium, the Society of Laboratory Automation and Screening, the Accelerating Therapeutics for Opportunities in Medicine Consortium, and she's also an advisor to Algia Therapeutics. Prior to UCSF, Michelle was Associate Director of Cell Biology at Synesis Pharmaceuticals, where she helped discover a protein interaction inhibitors for treatment for dry eye disease. I won't say the drug.
Lafitograsp, that's an easier one than most.
I'm wimpy. But maybe Michelle and Nancy, if you could start us off with a few slides just to introduce the audience to AmbAgon and the underlying science, and then Reshma and I will trade off peppering you with questions.
Yeah, thank you, Robert and Reshma. We'll put up a few slides to introduce AmbAgon, and we hope that talking about the founding of AmbAgon will demystify some of the path for those future founders out there who have great ideas. Let me get my slides up. Yeah. So AmbAgon is developing first-in-class drugs through targeted stabilization of protein complexes, protein-protein interactions. We are developing these molecular glues to stabilize protein-protein interactions between a specific type of a molecular chaperone protein called 1433 proteins and their previously undruggable client proteins to modulate the activity of those client proteins that are disease drivers. So 1433 proteins have this odd name. They were described in the 1970s. Because they're very abundant proteins and were described based on their position in 2D electrophoretic gels. So we have this rather prosaic name, 1433 proteins. The 1433 proteins endogenously regulate several thousand of these client proteins, many of which are involved in driving disease. Our initial focus is in oncology, but the 1433 proteins regulate disease-associated client proteins in a number of other therapeutic areas, including neurodegenerative diseases, inflammation, immunity, cardiovascular disease. The initial targets at AmbAgon for these molecular glues are undruggable transcription factors and difficult to drug non-enzymatic adapter proteins for patients with genetically defined cancers. And the company was founded by the leaders in 1433 drug discovery. There are three academic co-founders. Michelle here today, who is, as you've heard, the chair of the pharmaceutical chemistry department at UCSF, and has significant experience in biotech from her time at Synesis. Christian Ottman and Luc Brunsfeld are the other two academic co-founders. They are faculty at the Eindhoven University of Technology in Eindhoven, Netherlands. Christian is the world expert in 1433 proteins. He's been working on these for his whole career, and he's solved the great majority. He's a structural biologist and has solved the great majority of the 1433 crystal structures that are publicly available in the PDB. And Christian joined AmbAgon as our chief technology officer, while also retaining part of his academic appointment at Eindhoven. Luc Brunsfeld is the third co-founder. He is a medicinal chemist, also with experience in industry at Organon before he took his faculty position at Eindhoven University. And then I joined as the first full-time employee and the chief scientific officer during the seed financing. So AmbAgon was founded to solve a problem of undruggable targets that are involved in disease. And when we talk about undruggable targets, those are defined as proteins that have critical roles in a particular disease, but lack binding sites for drug molecules. I should say this slide was inspired by Robert, I think, who asked for a simple slide that illustrated what we mean by an undruggable protein. So Robert, we've used it extensively. Two challenges in undruggable proteins. One is proteins that are very flat and do not have structure that one can really attack with a small molecule. And then secondly, disordered proteins. Many of the disease-driving proteins are highly disordered, and again, those provide challenges for small molecule chemistry. The classic example here is transcription factors. There are a number of transcription factors that are known genetic drivers in many different cancers that have been very difficult to develop drugs for these reasons. And really, really hard to target directly. And so there's an opportunity to target these via protein-protein or protein-DNA interactions. So The advantage to 1433, the 1433 proteins are this elegant W-shaped structure that provide highly conserved binding sites for disordered proteins. So proteins with disorder bind into one of these two binding sites on 1433. There's a conserved phosphoserine, phosphothreonine recognition site on these client proteins. When those disordered portions of client proteins bind to 1433, 1433 is very rigid and now organizes the disorder into order. So converting spaghetti into a more ordered form that now one can develop a small molecule against. So solving both a potential flatness problem and a disordered problem. So binding to 1433 and these disordered critical disease-driving proteins allows us to target these disordered proteins. Now, binding of these client proteins to 1433 can have a number of different consequences in terms of the regulation of the client. And stabilizing with a small molecule molecular glue can then impact the subsequent function of those client proteins. In some cases where when those client proteins are transcription factors, there are several different fates as a consequence of binding to 1433 protein. For the case of estrogen receptor alpha, binding to 1433 restricts it into the cytoplasm, prevents it from entering into the nucleus and carrying out its transcriptional program, its oncogenic transcriptional program. For p53, binding to 1433 can stabilize destabilized 1433 mutations and, p53 mutations and restore p53 function. The tumor, so restoring, a gain of function in that case. For the MYC transcription factor binding to 1433, both retains it into the cytoplasm, prevents it from entering into the nucleus, and also confers ubiquitination and degradation onto that protein. So in each of these cases, stabilizing that naturally occurring interaction between 1433 and its client protein can interfere with that subsequent oncogenic or disease-driving function of the client protein. Now, the formation of AmbAgon was inspired by a natural product that has this stabilizing activity of stabilizing 1433 binding to client proteins that was initially identified in plants. At this point, I'm going to hand over to Michelle to talk about some of the foundational data that was generated by the academic co-founders.
Thanks, Nancy. Very nice introduction. I always smile when I see that transcription factor slide. So Nancy mentioned that there are thousands of proteins that bind to 1433, and that really 1433 is the shadow biology behind serine and threonine kinase phosphorylation. And so when we have all of these targets and all of this biology, what makes us think that we can make selective stabilizers? Because selectively stabilizing these interactions is really the key to the technology. So this confidence is provided at first by crystallography and then by data with small molecules. But if you look at the crystal structures that Christian's lab has solved, these are 3 examples that show how diverse the different protein-protein interactions really are. And these are peptides derived from the client protein in rainbow, bound to 1433 in white. And you just see how different those shapes are. So if you look at that pocket at the bottom, if you can advance the slide, Nancy, then you can see that those pockets have really different shapes and that different molecules are going to bind in those different shapes. So that's the natural product fusacoccin that Christian originally solved. It's a plant toxin produced by fungi. And you see how it binds in that pocket that's left open by estrogen receptor being a short peptide. Then you see these other types of molecules that bind preferentially to other types of interfaces. So we're binding at this rim of the interface. The shape of that pocket, that rim of the interface pocket, is really diverse from target to target. And therefore, the molecules that bind in those surfaces are really different from target to target. Then with YAP and TAZ on the other end, you see that we have synthetic macrocycles that bind to a completely different part of that surface, and they act more like a lid instead of like a blob of bubble gum that the other 2 surfaces fill in that pocket. Here we have a lid that closes over the peptide that stabilizes its binding to 1433. So because we have these really diverse chemical approaches that bind selectively to different types of interfaces, we have a lot of confidence that we can develop different types of drugs to different types of interfaces. And I want to just talk about one technology that, to us, it's one of the technologies that AmbAgon uses, but it was really key for demonstrating to us that this was going to be possible. So if you go forward. So the technology is called disulfide trapping. If you could go forward one more slide. One more, thanks. So disulfide trapping is this technology that we developed at Synesis that we use a lot in our lab that takes fragments, so tiny molecules bound to these disulfides in the middle, and we can screen them for binding to proteins that include a cysteine residue, near binding pockets. You can think of that cysteine residue as a fishing pole, and we can fish for things that bind in just the right location that also have the right geometry to make that disulfide bond. We detect those by mass spectrometry. So on the left, you see 1433 bound to estrogen receptor. And in that close-up, you see just the very tip of the estrogen receptor. That's actually the C-terminal residue of the whole receptor. And that big pocket, and in yellow, you see that fusacoccin is bound, can be bound there. And we have a cysteine residue, cis38, that's a wild-type cysteine residue positioned right at the rim of that pocket. So we can use that as our fishing pole to fish into that pocket. And if you click forward one more time, we can do the screen two different ways. We can screen for molecules that bind to 1433, but then we screen for molecules that only bind to 1433 in the presence of that peptide, of the R alpha. And that tells us that we're going to find compounds that stabilize that interaction. So we make a ternary complex that's specific to that particular peptide. And if you go forward one, we can see a couple of examples. So here's using two different types of disulfide trapping. One where the compound on the right is estrogen, left is estrogen receptor. We have that fragment bound to the protein. It contacts the peptide and it stabilizes the peptide protein interaction by 40-fold. Okay, so that's a really good starting point for initiating drug discovery. On the right, we can also tether off of the peptide itself. So some of these client peptides have cysteine residues themselves. And when we do this disulfide trapping off of the peptide, we see in this case, it binds into the same pocket with really a similar chemotype. And that all stabilizes that interaction by 5-fold. So if you go forward, these are just a couple of examples, but we've also demonstrated now looking at these different types of peptide protein interfaces that using the same library, so across the x-axis on these 4 plots is the same library screened against 1433 without any peptide bound. And then on the y-axis is the results for binding in the presence of those peptides. And you can see in the green boxes, those are compounds that don't bind to 1433 alone. They only bind in the presence of these peptides. In green, you see some compounds bind to all of the interfaces. They're really very avid for this interaction and kind of non-specific. And then if you click forward, I think, you see also that each interface has some specific scaffold that only bind to that particular protein peptide interface. And we can optimize both the non-selective and the selective ones into more potent stabilizers of these specific protein-protein interactions. So it's early in drug discovery, but for us, what was the key question? If you go forward now, the key questions were, how do you systematically stabilize protein-protein interactions? So there are lots of examples of stabilizing protein-protein interactions. Natural products do these, the IMiDs do these. We co-developed a compound called ISRIB that does this, but they were all discovered after the fact to do that. How do you do it prospectively? And so that's the technologies that we're really trying to develop. So by doing that, we're offering a really differentiated and exciting way to further this whole concept of stabilizing protein-protein interactions. And in our case, we're stabilizing native interactions. So similar to technologies you may know about, like PROTACs or molecular glue degraders, these bring proteins together and induce their degradation, but they are non-native protein-protein interactions. They're neomorphic interactions. At AmbAgon, we're stabilizing native interactions. So one advantage to this is we have a good prediction of what the biology should be because we're amplifying the native biology. Second, we have the potential to activate some targets, inhibit some targets, or degrade some targets based on that native biology. So in a sense, it's a more general approach than the degrader technology. And I think that's the last slide. So with that, we're going to open it up to questions.
Michelle and Nancy, thank you so much. And really, the point you made at the end about this being an approach to uh, use native biology and not just to grade, but also potentially to activate all in one platform. And that combined with really cutting edge fragment based discovery makes AmbAgon really pretty exciting. I wonder if you could, though, for us, you both had very long and distinguished careers already, but to take us back a little bit to when you first got excited about science, just briefly, what was inspiration that's launched both of your careers?
Nancy?
You want me to start? Depends how far back you want to go. So I grew up in Carmel Valley, California, kind of running wild through the woods and fishing things out of the river to take home and study. So that was sort of my first engagement with the natural world. But when I was an undergraduate student at UC Davis, I took a developmental biology class, and I was just hooked. I was fascinated by cell motility and how the cytoskeleton regulated cell motility and development. And that was really the first thing that hooked me into the mechanics of molecular cell biology.
Yeah, it's amazing. Yeah, I think, you know what Nancy said, going through the woods and fishing things out of the river to study, like children are born scientists, like why? That's all scientists do, right? So it's more a question of how did science not get drummed out of me? And that I went to college to, I don't know, maybe do some pre-med courses. And I took an art history class, and I got really interested in art history and art as a physical object and the chemistry that makes up a painting, and how you can understand its chemistry to understand how the painting has traveled through time, or other art objects have traveled through time. And so I was an art history and chemistry double major for a while. And that's what hooked me into it, is the symmetry. I studied inorganic chemistry, and a lot of paint pigments are inorganic compounds and crystals. And so understanding basically the physics and spectroscopy of how those things create color and how those colors change. And then when I went to graduate school at Caltech in chemistry, I took 3 classes in mineralogy, because that's basically before they get ground up into paint, their minerals. So it was like inorganic spectroscopy, and that's what got me hooked.
That's very cool. That's super cool. Actually, I was just down in Big Sur, drove through Carmel Valley. It is such a beautiful area. And I think, people who aren't in sciences see the analytical side of it, but there is a huge creative component to it. And I do like that idea that we're all born scientists. Another unique aspect of AmbAgon is that it's a multinational company with founders in different continents. Can you say how you originally came together as founders to create AmbAgon?
Yeah, I was at a meeting in Germany, a medicinal chemistry meeting in Germany. And Christian I think was the last year that he could win a Young Chemist Award. So he won a Young Chemist Award. And I gave a talk on protein-protein interactions, and he had known about my work a little bit too. So I saw his talk, and it was just so clear how exciting a concept that was. And I think I told him that he had sold his soul for such a great project. He could spend his whole career trying to understand and manipulate this target class. And he said, his dream was to start a company to do that. And he'd already had the name picked out, and could I help him? And so we finally, took us a little while to find somebody who was crazy enough to move over to the United States from the Netherlands for 6 or 7 months. And Alina Sebesma came over as her master's project, and got this project started with us. And then she became the first employee after Nancy.
Oh, very cool. The purpose of this seminar series with Y Combinator and NBC BioLabs is to really inspire others to start companies, but also to show them the path, or at least show them, your path in this particular case. And so AmbAgon's won, I think, 4 pitch competitions. You've won 2 golden tickets. And for people who don't know, a golden ticket is a free lab bench for a year at NBC BioLabs. It's awarded by our pharma partners. So they can be quite helpful in companies getting started, both in terms of providing a free lab bench and then also some big pharma validation. Can you or Nancy or both say a bit about how those competitions, how the golden tickets helped you and helped AmbAgon get going?
Yeah, I guess I should take that one, Nancy, because some of it predates Nancy.
Yeah, before my time.
Yeah. So the first one was we were just getting the company started and putting together white papers, thinking about what targets we wanted to go after first and what therapeutic areas. And we got a notification from Catalyst program at UCSF about the Science to Startup, which is a pitch competition in Boston. So we were applying for that, and about that same time, the golden ticket came up, and so we sent the same slide deck to both. Like, let's see what happens. And about the same time, we found out that we were invited to the next level of competition at Science to Startup, which included putting together an official pitch deck, and that came with people who could help us put together that deck. So it came with a suite of consultants. And that we found out we were also invited to pitch to the Mission Bay Capital golden ticket. So I called the Science to Startup people. I was like, can you help me put together the pitch deck for the golden ticket? And they were really excited and really did a great job helping us. We reached out to some people that we knew who were already in the BioLabs, people who had been a grad student and postdoc in the lab next door to mine, and they had won a golden ticket. And so I asked them for help, and very generous, giving us their slide deck and some advice. So this whole, takes a village, this whole international network helped us put together that pitch. And that was to a big pharma venture group. And the head of oncology discovery was also in that pitch. And he had worked in protein interactions, super excited about it, got it right away, and has been a great champion ever since. He's now on our scientific advisory board. And they were the first group to really sign up with us and to help us get funded. And it took a while to get together the consortium because it's complicated and international. Nancy we could talk about that later. But they were really with us all the way. So I'd say that winning that pitch competition, the golden ticket, was really when we became a company in a sense. And then we won a second one because, because 2 benches is better than 1. And that was also a really great experience. And pitching at Science to Startup was another great way to meet a lot of venture capitalists on the East Coast, since that was in Boston. RA Capital hosted that. And in the end, RA Capital also, became a big champion of AmbAgon. The fourth one was in the Netherlands. So the Dutch Venture Challenge that Luke spearheaded. And that caused us to put together a really nice white paper that we kept editing and circulating throughout the process. So, it's all about meeting people and getting your story out there and improving your story through exposure.
Zero in on the timeline a little bit. Like, is the golden tickets were before you incorporated and started the company, or what was the sequence?
Yeah, they were way before.
Before. Okay.
Yeah.
How many years?
Well, a year.
A year
Before.
But when I look back at slide decks, when I look back at your slide decks, I see how the input that you got from winning those competitions refined your story, right? Refined the AmbAgon story from those very initial. Some of the targets changed, how you told the story changed. So it seems like each of those pitch competitions gave you valuable input into developing the story that eventually got funded.
Definitely, just like you're saying, where Robert said, okay, I need a slide early on that says, what's cool and hard about transcription factors? So just like make it easy for me. And yeah, it's true. And if you're not open to that feedback, it's going to be a lot bigger slog for you. No. So
would you
I'd say take advice
to a like scientist who's in academia to like enter their ideas regularly into the competitions and try and get some feedback? Is that like a good path in general, or is it like really if you're clearly on a trajectory to start a company?
I think it depends a little bit. So the Science to Startup, for example, is very competitive. So you need to be serious enough to be competitive, but it doesn't come with a timeline. I think the golden ticket is really for a bench. So you should be pretty sure that within a year you're going to be using that bench. And the pharma company, which I think they didn't want to be named, so that's why I'm not naming them, but the pharma company let us delay until we were ready to use it. Astellas, the second golden ticket, said you have to use it by a certain date. Even during the pandemic, the time wound down. So everybody has a different timeline. So you want to use one that's compatible with your timeline. So entering competitions is definitely a good way to learn. Also talking to the sorts of people that would be in those competitions, right? And you don't need to be afraid. So I used to live in Los Angeles, and we had a lot of friends who were in the business, right, in Hollywood. And they said, a producer will always talk to you because they don't want to be the ones that didn't talk to you. And venture capitalists are the same way, really. Right, Robert? I mean, right, Rashma? You'll talk to people if they have a good idea, especially if they're from a reputable, background, because you want to help and because you don't want to be the one who didn't hear about it.
And you never know. You never know.
FOMO.
The good ideas are the strange ones oftentimes.
I feel like we're being called out, Robert. No, I'm just kidding. Tell us about that moment, like when you decide to incorporate the company, like what was the ingredients that, first of all, the IP that went into it and started the trajectory of the company as AmbAgon?
We have some foundational IP that's really around the applications of the technology and some of the things that we learned. But the company, I think you can tell from that was the data that we had when we started the company. We didn't have composition of matter claims yet. And so it's really built as a platform company. And that comes with certain joys and challenges. There are some venture groups that aren't going to be interested in something that's that early. But the ones that are, they tend to have big vision. They tend to have a lot of science interest, and they tend to be good partners. Like they want to help you partner. I just saw in the chat that KT said that we can say it's AbbVie. So it's AbbVie Ventures. And so they're very scientifically engaged too. And another one of our investors said, we love your technology. We love you guys. We want you to add these targets. If you add these targets, we'll invest. So So
have you already made like some of those, I guess the synthetic microcycle cap, like have you already made some of those and tried them in experiments, and like what was the result?
Right. So we had some cell data and we had some in vitro data. So we felt that we had crossed a really important conceptual hurdle. So you never know, right? Drug discovery is a series of hurdles, and you don't really know which ones are going to be the high hurdles. And this idea of fail fast, if you hit a high hurdle early, if all the hurdles after that are going to be low or average, you don't want to, you just need to know you've crossed the highest hurdle. And so for us, it was how to do it. Can we do it with drug-like molecules? And so seeing that we had that data for us was important reason to believe. And I think that also for the venture capitalists, the ones that could see, all right, that's not the drug, but I can see how you can get from there to the drug.
Got it. Yeah. So cell data with the compounds that were naturally existing or demonstrated the concept.
Yeah, so we had cell data and animal data with the natural products. We had cell data with macrocycles and biochemical data, but selectivity data with fragments. Nancy, what struck you?
What I remember is that what you told me was there was sort of a moment when you three academic founders all agreed that there was enough information out here. This was a really interesting idea, and if anybody was going to do it, you wanted to be the ones that were going to do it, and that was really, really a lot of what drove it. Right, realizing this is something really special. We have the proof of concept. We have the expertise, and we should do this before somebody else does.
And we need somebody to select the right targets, and that's why we hired Nancy the second we met her.
And you mentioned that you had the data. Did you have any like IP provisional, or what did that come out of the university? Like, what was the IP story?
We have one patent that's joint with 3 different groups, with our university, Eindhoven University of Technology, and a CRO that co-developed some of the compounds with them. So it's a 3-way patent, and we have an option. AmbAgon has an option in this negotiating the license.
And so, like, transitioning a bit, after the incorporation of the company, and remind me roughly when was that, like in time?
In the middle of the pandemic. Oh, the seed funding closed in the middle of the pandemic in July.
In July.
July of
Okay, so
2020.
you have your patent option and your animal data and your cell data, and kind of went to go raise around. What did that first money look like? Can you tell us a little bit of story of what happened then in July?
Well, July is when the seed funding came in.
Closed. Yeah.
Yeah,
Okay,
closed. So
maybe
we'd been
started.
talking to them for about a year. I'd say we had, and the syndicate hasn't been disclosed publicly, we haven't done a push. So we'll be sort of vague, but there are corporate venture, institutional investors in the US and Europe, and specialty investors. So it's a range of types, range of geographies, and in a sense, maybe that gives them a range of value propositions. And so it took some time for it to really gel into the syndicate. But we had a lot of those players being interested for quite a while before. So I think the lesson there is just stick with it. Just, things are always slower and more complicated than you think. And
yeah.
Yeah.
I mean, 1 year to kind of get all of the materials through. Did you talk to them regularly throughout that period, and then they're like, okay, this has reached the maturity to invest, or how did that discussion go?
I think Nancy probably have another perspective on this too. It seemed like we were, they were talking to each other, we would talk to them. It's just a lot of people, and so just a lot of talking and across time zones. And so there's 3 hours in the day that everybody could talk, and so I think that's also part of what dragged it out. It's just
and you founders had a couple of key relationships, but you didn't have a designated lead investor to then go out. You founders put the syndicate together yourselves, essentially, with some lead like behavior from a couple of those early committed investors, right?
Yeah, that's a good way to describe it.
Sometimes there's a typical path, right, where an academic founder takes an idea to a venture fund and they then take the lead in assembling the syndicate. This was a little different.
Right. And so it was always done with their help, but it's true, we did do a lot of the sort of bringing people together also. And in the end, the investments there isn't a lead investor financially either so much in the seed round. It's a lot of groups wanting to contribute. And then I think in the Series A, you'll see a real lead investor will be much more typical in the seed.
You're sort of touching on it, but I wonder if you could say explicitly, are there things that you know now that you wish you had known when you started?
I think trying to convince them to assign a lead investor would have made it faster. Yeah.
I was going to say how long it would take, right? The mechanics.
Yeah.
So how long it takes to get the commitments for the funding that you need. And it's a lot of repeated conversations, a lot of discussions, and then long path from commitment to close.
And I would also have brought in a business person earlier. So we brought in a business person, and we looked a little bit and we tried to get some help on that, and we didn't get we didn't bring in a business person until after we had some early term sheets, until we had a lot of the syndicate players around, and then trying to bring in somebody. And those people can be, they make their living being pretty aggressive. And so there's just what is this new culture dropped into the mix? So I would bring a business person in earlier so that they're always part of the discussion. I think that would have sped things up a lot too. Plus just experience.
Yeah. This is one of the things that we sometimes see is surprising to founders. They're like, oh, fundraising, seems difficult and takes a long time, even for folks who have won several golden tickets and who have a lot of momentum with their science. So just something to be aware of, but also not unovercomable. I respect for being so persistent with it and closing your round in the middle of the pandemic. Was there challenges to having lab benches in different continents with regards to getting the company up and running and scientifically?
We started on Zoom, but we started at a time when the whole world was working on Zoom. So in a way, it normalized it, right? And
Yeah.
we'll always be on Zoom because of our geographical distribution.
Right.
So it was not so difficult to start by Zoom. I think many companies worried during the pandemic about retaining culture during that time. We were really thinking about building culture. So a slightly different strategy of how do you build culture at two sites that are 9 hours apart during a pandemic when you can't even travel back and forth, right? But a lot of our culture is empowering our scientists, and working remotely on Zoom certainly, forces that. And our scientists have really taken off and run with their different responsibilities as a consequence of us being distributed and being on Zoom.
Yesterday, Nancy mentioned that, on the one hand, we have 3 hours a day that we can have meetings. On the other hand, we have 6 to 8 hours a day where we don't have any meetings. So that's can also be really helpful.
Yeah, the geographic differences helped us that way, right? We have time for focus work and lab work. All of our all company meetings have to fit into 2 or 3 hours time, early Pacific time.
That actually sounds like it could be a strategic advantage. That goes great.
We also, I think the other advantage, it's quite challenging to recruit talented scientists right now. And so being able to recruit from Europe as well as from the US, I think definitely gives us an advantage.
There was another founder CEO that I was speaking with recently who was talking about fundraising, and he said he's experienced, he's done this before. He said, as you know, it's slow until it's fast, and that's often the case. I've heard, Reshma would know better than I, in the tech space, that deals can come together very quickly. But in the biotech space, it's very typical for it to take a long time for financing to come together. There's cooperativity, and then sometimes even in the case of AmbAgon, where you have a remarkable platform where everyone could see all the potential, you still, at least on the investor side, have to build the real use cases and say, these are the first couple of targets that we anticipate going after. And so it just takes a lot of time to both understand what those targets are, and then also get the investors to come around and understand that there is potential there. But we've touched on sort of some of the challenges, putting the fun in fundraising. But besides fundraising, what makes entrepreneurship fun?
So, so many things. One is just the scale. If you stay in academics, this is also something that we all have to decide. If we're biotech savvy or biotech interested academics, do you jump ship? Do you go to the company? And this really came up for Christian. Like, this is his whole research program could be in this company now. Does he close down the research lab? And what are the advantages of staying close to the academic space? And so for me, it's on the opposite side. We want to do things in 1433 that are help AmbAgon tell their story without being interfering with AmbAgon's story. And so that's fun. I think the different pace and the different value propositions really that you have in these different environments is really fun and exciting.
And I think from the AmbAgon company side, both fun and scary in a startup is you are in control of your destiny. Right? So huge opportunity to drive the company forward and really build that value, but huge responsibility comes with that. It's really exciting.
Yeah, and on this side, we're always in awe of the founders and entrepreneurs that take the leap, because it is incredibly fun, but it is a lot like jumping out of an airplane. You've got to believe in what you're doing and believe that your parachute's going to open and it's all going to be okay.
If I could just say one thing about bringing Nancy on, that I really appreciated from her career, it became really clear that she was very mission driven, that she wasn't going to stay in a job that was comfortable and not exactly what she wanted, or she wasn't really having the maximum impact. So bringing those first employees in too, and just building that team around, like, what's your vision? And can you bring people in that can amplify your vision, and correct your vision? is also a really fun and exciting thing, getting to know people that are, spectacular, that are just not people you'd have met otherwise.
Well, thanks for doing it. Rishma, I think we'll have some audience questions now.
Indeed, we do. So from Beatrice, are you accessing only hydrophilic proteins with 1433?
Well, they're all phosphorylated and they're all cytoplasmic or nuclear, so they aren't transmembrane proteins. I think that's what that question means. Although, correct me, Beatrice, if I'm getting it wrong. Some of the targets are transmembrane proteins. So, for example, CFTR, the ion channel that causes cystic fibrosis, is a 1433 client, and a lot of ion channels are trafficked to the surface, so they're activated by 1433 binding. So that's thanks, Beatrice. Yeah, so that's one of the interesting things too, is activating the ability to activate an enzyme or a non-enzyme protein is pretty unusual.
Any advice for biologists without a chemistry background on how to transition into developing candidate targets? From Odessa.
Yeah, I think if you're if you are a biologist and you're working within a company setting, right, there are opportunities to get exposure to candidate development, I think. I don't know if we're talking about identifying targets to enter into drug discovery programs, or if we're talking about actually advancing a compound towards the clinic. I think for the latter, that's really a skill learned on the job. You can't take a class in it, right? And it's part of what makes this business so exciting, is that each molecule is different. But really, the opportunity there is to learn through exposure on project teams and within companies where that's happening.
I think another aspect. Oh, I'm sorry. Another aspect to that question is that I work with some companies, or just know some companies where the biology is really amazing, but they really don't have any drug discovery experience. So how do you know that your target identified through genetic means is druggable? And it is very helpful to bring in somebody who's done some drug discovery, but who isn't going to say no too quickly, who's going to think about how you can drug that target, or how you might think about drugging that target. Yeah, it is a separate set of expertise from biology or chemistry, really.
Is it recommended for someone who has no specific experience in advancing those candidates, like through a process, like to do it as a startup on their first try, or does experience really help? What's the thought on that?
Experience
is way more important than knowledge. It's like...
But if you have experience, I think inexperienced founder can benefit from experienced advisors, right? And that works.
Yeah, and bringing in consultants. There's a whole ecosystem of people to help you, and groups like Mission Bay BioLabs and Combinator will help put you in contact with those experts that you need to hire or bring in. Okay, I see a bunch of
questions,
things in the chat. Yeah, awesome.
We have actually, if you don't mind, Mark Schumann's joined, and he wanted to ask a question directly. At least I believe he did. He raised his hand. So if we could allow Mark to ask his question, that'd be great.
Hey, Mark.
I made a mistake. Sorry. I'll let someone else who has better questions.
Good to have you on board, though, Mark.
No worries. That's the learning curve of Zoom, everything. But so in the abundance of targets for the 1433 proteins, is there potentially a hurdle for the drug to have a biological effect?
Oh, hey, that's a question from Michelle. Hey, Michelle.
Michelle.
So a good question. 1433 proteins are one of the most highly abundant proteins in the cell. So the level of 1433 is probably not a limitation. And in that case, I think then the target that you're stabilizing may become a limitation. So very, very abundant targets may be a challenge to fully stabilize. But the great majority of the targets that we're interested in, 1433 is in high abundance relative to target.
So one of the things, this is a question from Doug, one of the things that Doug was impressed with when watching AmbAgon mature was how much progress was made in the target selection. Like, did your fundraising process inform your target selection, or did you have like your own thesis on it? What was the process for target selection?
It did.
Yeah. Yeah, there's always this balance. So when we selected targets in the academic lab, it wasn't because they were the best drug targets, it's because they were the best targets to test a hypothesis about the technology.
Mhm.
And they were reasonable drug targets. I mean, they're good enough drug targets that you can publish a paper and call them, or write to NIH and call them drug targets, and you'll be believed. But it doesn't necessarily mean that they're the best targets to pick up in terms of competition, feasibility, clinical trial design, the kinds of things that the CSO and CEO really need to be focused on in target selection. So I think we had some discussions early on where people, I might say, missed the forest for the trees, that they saw the example and they said, well, we don't want to develop that target, so we're not interested in your company. So yeah, so that was part of the refinement of the story, is convincing people that with this data, we could develop drugs for these other targets, and then what those targets should be. And then Nancy, you want to amplify that?
Yeah, no, I was just going to say that, yes, the initial target that you started with, I agree, people focused on the target and, a clear cancer driver, but one that's really a very small unmet medical need at this point, right? Many drugs against this target. And you had some strong input from certain investors that had targets that they were interested in, that you included into the portfolio. And so I think having a handful of well-vetted targets that were the commitment to the investors, that these are the ones that we're going to start the company on, I think was an important part in the funding.
Yeah, that they could visualize wanting to do that and why it wasn't to solve the problem otherwise.
Right.
And yeah, and that is a challenge, I think, with a platform company, is balancing that, because we really didn't want to be known as, the 3 targets that she had on that slide. Was it Nikki or off of P53? You could get excited or slam any of those targets for any number of biological reasons. So we didn't want to be the company, but we needed to show that if we could, the balance of the platform and the targets is really key.
Interesting. It sounds like, first of all, that you developed a rubric, right? Like, this is the clinical need, and this is the clinical trial design, and this is the kind of potential of each of the targets. And you use that as a communication device to get investors on board. Did I interpret that correctly? Yeah.
Yeah, we've refined that quite a bit, post with company formation. We've really made that a more sophisticated process for assessing targets.
But I'm sensing that you don't want to be like really heavily associated with your top targets in the long term. Is there kind of a thinking along those lines about why you yourselves want to have a little distance from those targets?
Well, when they're successful, we absolutely want to be associated with them. What we don't want is if we take the world's hardest target nobody's been able to solve for 50 years and we don't solve it in 18 months, and then they don't invest us anymore. That's where the balance comes in. That we've learned something, or we've built the company that if this target doesn't work, target number 2 is just as cool. Target number 6 is just as cool, right? So part of that prioritization that Nancy's developing, and Nancy and her team are developing, is to make sure we're learning and not working on something that's not going to work, right? And continuing to have good ideas. Does that make sense?
Yeah, kind of like a triage, and you're keeping people informed about what you've learned.
Yes.
And do you think that part of the, when you raise your next round, do you see those targets being fixed, or they're still in flux, or how do you think about the progression of the company that way?
Right. We have a continuous flow of targets through our assessment and validation, right, and then into hit finding. And the key, so there are many potentially interesting targets with 1433. That's part of what attracted me to the company. It was many of these classic undruggable proteins that can be targeted. And so the key for us is to do those assessment and validations thoroughly, and then make data-driven decisions on which ones go forward as we start generating hits and digging into the biology of how these client proteins interact with 14-3 and what the consequences are on the disease.
I think that also had something to do with the funding strategy, which went both ways, right? It's what we wanted to have funded and what other people wanted to pay. But calling ourselves a seed it's a pretty large seed as far as it goes. So why call it a seed? Because I had this feeling that during a seed, you can be developing platforms, you can be developing a target technology, you can be developing hits and leads. And once you have a Series A, it's how many months to the clinic, how many months to the development candidate, how many months to the second one being a development candidate. And we wanted to have some money and time to select the best targets and the most feasible targets and the most exciting targets. So calling that a big seed instead of a smallish Series A had that kind of expectation setting in my mind. I don't know if it really worked or made any difference. I don't know, Robert, you're an investor. What do you think?
Well, as Doug sort of asked the question, I think the progression in thinking about what the right first couple of targets was really critical for us on the investment side. I mean, we're really obviously impressed with the platform and you initially, but we really, from an investment thesis standpoint, had to know, what could you really drug with this that could be a successful drug. And seed versus Series A, actually, it doesn't much matter. We really do need it to be at least thought through. Those plans can change based on the data, as Nancy was saying. But the initial path really does need to be thought through, whether it's a seed or Series A.
It does, I think, like crystallizing the opportunities, like it does help get people anchored into like what things could be, even though in your case, there's actually lots and lots of opportunities. And a big part of the job is filtering them and making sure that they actually have that potential. We are actually almost at time. Is there a lightning version to what about the Ras pathway? I'm not sure what a Ras pathway is.
Yeah, so if you type into QIAGEN's website where they give you whole pathways and you type in growth factor receptor signaling, you'll get the growth factor pathway that the person's talking about. If you type in growth factor receptor pathway 14-3, you'll get the same pathway with 14-3 on it 18 times. Okay, so that's what I mean. It's a shadow biology, and it's really key in the Ras pathway, particularly in CRAF and BRAF activation. And several cryo-EM structures have come out in the last couple of years that really highlight both the inhibiting role and the activating role, 14-3 and Ras pathway. So that's a great example of biology, what's great biology versus what's a great drug target. And is a RAF inhibitor a great drug target, or are people going to say we have a lot of RAF inhibitors already? So that's part of, I think that's an excellent example of where it's great science, and we probably have to push it further for it to be something that a company would want to pick up to demonstrate the differentiability.
We are almost at time, but thank you so much for sharing your story today and so many like vivid details about what it's like to start a company in this space. So we really appreciate it. For those in the audience, like we're really encouraging folks to take the first step in starting their company. Robert just shared the NBC BioLabs website here, and you can always reach me via email here as well. And we hope to, in the next few years, have some who are in the audience talking about their story as well. And thank you again to Michelle and Nancy for taking the time to speak with us today.
Thanks so much for the invitation.
Thanks for the invitation.
It's great to talk to you.
Thank you so much, everybody. Take care.